So for our second speaker of the session, we now welcome Dr. Jack Brandt from the Swedish University of Agricultural Science in Sweden, who is going to talk about how pharmaceutical pollution alters river-to-sea migration success in Atlantic salmon. So Jack, you can share your screen.
I think the presentation's been recorded, but
sorry.
yeah, no worries. But no, thanks for the introduction, Catherine, and nice to virtually meet everyone. I just wanted to flag quickly before my talk as well that if you're interested in this type of work, I've also got a similar poster on a similar sort of topic that you can find through the relevant links, etc.
Okay, so it was my job to share the screen in the end. So I will now play the recording. Thank you, Jack.
So hi everyone, my name's Jack and I'm a researcher at the Swedish University of Agricultural Sciences. Today I'm going to be talking about some of the work that I did here as a part of my postdoc, investigating how pharmaceutical pollution can alter the seaward migration success of Atlantic salmon. But I'd like to start today by emphasizing that this project was a huge team effort, including Dr. Jake Martin, who's a fantastic researcher that has designed sort of many of the graphics and slides that you're going to be seeing today. So as a bit of background, the contamination of ecosystems with synthetic chemicals is now considered the fastest growing agent of global environmental change. Now this includes things like pharmaceutical compounds, of which almost 1,000 different drugs have now been detected in aquatic systems worldwide. And worryingly, the problem is getting worse. Indeed, the number of different pharmaceuticals detected each year is escalating, and they're turning up across the globe, including in places like Antarctica. So how exactly do these products make it from the pharmacy shelf to the environment? So typically it's through usage and excretion, whereby a proportion of the drug taken will be extracted by the body unchanged, or as an active metabolite. From here, these products then make their way through the wastewater system. The wastewater system is not really designed to deal with these types of pollutants, so many of them are not effectively removed, thus resulting in many of these pharmaceuticals being pumped into the environment. Now, while pharmaceuticals are often detected in our waterways at relatively low concentrations, these chemicals represent a unique challenge for ecotoxicology because they're often designed to alter specific biological functions and to be effective at very low doses. Indeed, many of the targets of pharmaceutical drugs are what we call evolutionarily conserved, meaning that they're also present in a wide variety of non-target wildlife species. And as a result, these drugs have the potential to affect a range of important biological traits and functions that are not typically assessed in ecotoxicology. One particularly concerning class of pharmaceuticals are those psychoactive compounds that alter human brain activity and behavior. So things like antidepressants, anti-anxiety medications, and painkillers. And indeed, around 13% of all detected chemicals in European rivers are thought to have some neuroactive potential. And indeed, much research over the past 2 decades has shown that exposure to these psychoactive compounds can alter the behavior of wildlife species. And now, given the importance of animal behavior to survival and reproduction, such effects could have significant implications for the persistence of wildlife populations. So recently, as part of projects led by Jake Martin, we conducted a systematic search of the behavioral ecotoxicology literature to find what the key research gaps are in the field and what the barriers in translating this research into effective policy are. And so we found that after exposure to some pharmaceutical compound, around 78% of studies in this research area test the behavior of individuals by themselves in social isolation. Similarly, almost all of this research is done under artificial laboratory conditions that sort of fail to capture real-world complexity, often assuming that sort of drug-induced changes recorded in the laboratory likely affect organismal performance in the wild. But we know that environmental conditions, including social conditions, can significantly influence animal behavior, emphasizing the need to conduct more sort of ecologically based research. And finally, the pharmaceutical concentrations tested in these studies are on average around 43 times greater than those actually observed within the environment. So we set out to investigate whether exposure to environmental levels of psychoactive pharmaceuticals actually alter animal behavior in the wild and determine what consequences this may have for populations. So to do this, we focused on two compounds. Clobazam, which is a benzodiazepine drug commonly prescribed for mood disorders and epilepsy, as well as tramadol, which is an opioid drug commonly used as a painkiller in human patients. Now, importantly, both of these drugs are routinely detected in aquatic ecosystems around the world, including in areas where they co-occur together. And interestingly, both compounds are known to have adverse chemical reactions if prescribed together in human patients, meaning that wildlife that are simultaneously exposed to both compounds may suffer even greater effects. So for our study, we used 2-year-old Atlantic salmon. Now, this species starts its life in fresh water. After around 2 years in our system, young fish migrate from the rivers to the sea, where they grow and mature before returning to their natal river to spawn. But importantly, salmon populations have suffered substantial declines in recent years, largely owing to anthropogenic environmental change, with chemical pollution being identified as a contributing factor. Now, we exposed these salmon to one of 4 treatments using novel, long-lasting pharmaceutical implants developed by my collaborator, Ellen McCullum. Salmon either received a control implant with no drugs, an implant containing clobazam, an implant containing tramadol, or an implant containing a mixture of both clobazam and tramadol. So as a first step in our study, we wanted to verify that the drugs present in these implants were actually entering the fish's bodies at environmentally relevant concentrations. So we took around 260 fish and divided them equally amongst the treatment groups. So fish were held in large flow-through tanks that received constant fresh water from the local river. Now, 4 fish per treatment were sampled at each of 7 time points between 1 and 30 days after implant exposure. Now, multiple tissue samples were taken from these fish, but today I'll just be sort of focusing on the brain samples just for brevity. So I don't want to spend too long on this result, but essentially we found that the levels of clobazam in the brains of clobazam and mixture-exposed fish were around 5 nanograms per gram, while levels of tramadol recorded in the brains of tramadol or mixture-exposed fish were around 2 to 3 nanograms per gram. Now, the absolute values aren't really that important here, but what is important is that these concentrations are similar to previous studies which have sampled fish from contaminated ecosystems in the wild. So now that we've validated our chemical implants, we wanted to conduct a field study to investigate whether these pollutants can alter animal behavior, movement, and migration success in the wild. So we took almost 280 Atlantic salmon and split them equally amongst the same 4 treatment groups. So we replicated the experiment across 2 years to minimize the effect of natural yearly variation in migration conditions. And prior to release, all fish were implanted with acoustic telemetry tags. Now, if you're not familiar with acoustic telemetry, this technology is widely used in aquatic tracking research and basically involves performing a small surgery on the fish. So during this surgery, a small transmitter tag is inserted into the body cavity of the fish. Now, these transmitters are programmed to emit a unique acoustic signal when the fish is in the water. And by using specifically placed microphones throughout the river that detect once these transmitters come into range, we can essentially see where and when the fish is moving. So after tagging, all fish were released into the River Dal, or Dalälven in Swedish, which is located in central Sweden. And they were released around 28 kilometers upstream of the Baltic Sea. Now, along the river, we had placed around 40 acoustic receivers. So they're sort of microphones I was talking about just before. And this was done in order to track the fish's migratory journey to the sea. Now here I'm going to show you the relative migratory success of the 4 treatment groups. So these plots represent the number of fish that passed each of these checkpoints along the river. So here you can see that just a little downstream of the release site, there were no differences amongst treatments in the number of fish passing this point in the river, which is to be expected considering that we released equal numbers of each of the treatment groups. However, at around halfway along their migratory journey, you can see a clear effect of treatment emerge, whereby clobazam-exposed fish are successfully passing through this checkpoint in greater numbers relative to all other treatment groups. And this treatment difference was maintained until fish reached the sea, with approximately twice as more clobazam-exposed fish reaching the Baltic Sea relative to all other treatment groups. And so we found that twice as many clobazam-exposed fish ultimately completed their seaward migration relative to all other treatment groups. Now, like I am sure is true for most of you, our next question was, why was this the case? So the eagle-eyed observer may have noticed from earlier that there were two hydropower dams located along the river that fish had to pass in order to reach the Baltic Sea. Now, hydropower dams are known to significantly delay downstream migration and can result in increased predation threat at these sites, as well as greater energy expenditure. So we wondered whether clobazam exposure may have been altering how fish interacted with the dams and whether this ultimately caused their heightened migration success. So here you can see the time taken to pass through the dams on the y-axis, with higher scores indicating slower fish and lower scores indicating faster fish. And as you can see from the first dam, that again, clobazam-exposed fish were around twice as fast at passing the first hydropower dam in the river relative to the other treatment groups. And this story was similar for the second dam in the river, with again, clobazam-exposed fish being faster than the other treatment groups. But just to caveat that for this dam, we only had data for one of the study years due to the lack of a direct downstream receiver in the second year. So we'd found that clobazam-exposed fish were around 2 to 3 times faster at passing the dams along their migration route. And we suspected that this may be causing, or at least partly contributing to, the differences in migration success that we were seeing. But like I presume you all are, we also wondered how clobazam was actually causing this increased dam passage speed. Now, it's known that anti-anxiety benzodiazepine drugs like clobazam can reduce physiological stress in fish, which can then alter risk-taking and social behaviors. Now, interestingly, these behaviors are thought to be important for downstream salmon migration. So we followed up on our fieldwork with a lab experiment to investigate whether these concentrations of clobazam could be altering, risk-taking and social behavior in Atlantic salmon. So we took around 130 salmon and exposed them to either a control implant, a low clobazam implant, which was the same concentration that we'd previously used in our field study, or a high clobazam implant, which was approximately twice the dose of our low treatment. So within these treatment groups, fish were randomly allocated to groups of 6 individuals. And within these groups, salmon underwent 6 trials of social and risk-taking behavior, both in the presence and absence of a common juvenile salmon predator, the northern pike. Now, we measured a few behavioral traits from the trials, but today I'll just be talking about the average group area during the trial, which is a common measure of fish social behavior. Whereby smaller group areas indicate a more cohesive social group and vice versa for larger group areas. Now you can see here group area plotted on the y-axis, again with higher scores indicating less social groups, lower scores indicating more social groups. And you can see that in the absence of a fish predator, there were no substantial differences amongst our treatments in their social behavior. However, when the predator was present, similar to how conditions would be in the wild, and specifically around dams where predators are known to aggregate, particularly during the salmon seaward migration period, we found that clobazam-exposed fish were much less socially cohesive than the control groups. So we'd found that clobazam-exposed fish were less social and somewhat more solitary than unexposed fish, and that this occurred in the presence of a fish predator. But how does this relate to dam passage? And so interestingly, previous experimental research has demonstrated that solitary fish pass downstream barriers up to 23 times faster than those in social groups. Now, the authors of this study suggested that when in groups, group members match each other's orientation to maintain their school structure, leading sometimes to collective upstream swimming and slower eventual passage at these downstream sites. So from our work, at least it seems initially, that clobazam exposure reduces social cohesion, particularly in the presence of predators, like would be the case when passing dams. This reduction in social behavior subsequently leads or contributes to increased dam passage speed, and this also contributes ultimately to greater migration success in these clobazam exposed fish. But is this a good thing? And so I think it's really, really important to emphasize that that even seemingly beneficial behavioral changes, like faster passage through barriers, may come at a cost. So altered migration timing, for instance, might lead fish to arrive at sea when conditions are suboptimal, or expose them to new predators and risks when completing their migration. And over time, these subtle shifts could influence population dynamics and even ecosystem balance. So if you're interested in these results and want to dive into them in a little more detail, please check out our recent article in Science, which you can access through this online QR code. And finally, before I finish up, I just again want to thank all of my amazing collaborators who worked so hard on this project, and of course, all the funders that made it possible, and even ecosystems worked so hard on this.
So thank you for this pre-recorded talk. I'm leaving this slide so people can have the reference.
Perfect.
So we already have some questions in the chat. I will read them to you. First of all, Laure says, congratulations about your science paper. It's not a
Thanks.
question, but
Yeah, it was a mammoth effort and probably something I don't want to undertake anytime soon. So, but no, I appreciate it. Thank you.
Then David asks you, what change would be needed to the way that we treat sewage water to remove the drugs? Is it even possible, or do we need to change the way we use them?
Yeah, that's a really, really good question. I think so essentially there's lots of different ways that we can try and remove these things from the environment. So there are certainly sort of advanced wastewater treatment techniques which can sort of remove a substantial portion, but likely not all of these substances from the environment. And again, this is very context specific. You know, it depends on the specific substance we're talking about, etc. I think it's important to note that while some of these more advanced wastewater treatment technologies are available in high income and high GDP countries, in many parts of the world, they're still not really available. And so one of the other things that our research group has been sort of writing about recently and has an article on is this concept of sort of greener pharmacy, designing greener drugs. So that's sort of the idea that, you know, during drug design, acknowledging that a portion of these things are going to end up in the environment. And so how do we design them from the outset that they sort of either biodegrade quicker or don't cause harm to non-target wildlife species? But it's a really, really good question. And essentially, there's lots of different things we can do.
Yes, different approach, but the best way would be to, from the start,
Yeah,
fix this issue.
yeah, exactly.
So then Laura is asking, I wanted to ask, what is the normal concentration of this drug in the wild compared to the concentration you used here? And if there is a big difference, do you still expect an effect under the normal concentration?
Yeah, it's another really good question. So we specifically chose the concentrations used in our experiment to mimic what would be seen in contaminated ecosystems in the wild. So usually in wild systems, concentrations of these drugs range from the low sort of single digit nanogram per liter to anywhere up to maybe 100 to maybe at the very, very highest end, 1,000 nanograms per liter. Now, of course, not all of that is absorbed by an organism and gets taken up into, you know, their brains and affects neural systems and things like that. But in wild systems, you usually see these sort of low nanogram, tens of nanograms per gram in terms of how much is in their actual brain. And so those concentrations were sort of right in line with the concentrations we used in our experiment. And so we specifically chose them to sort of mimic those natural conditions that you would see.
Okay, so then Tarun, he asked two questions. While using fresh water from river, did you check presence of chemical pollutants in the water and its effect, or it didn't matter as it's some sort of control noise across all treatments?
Yeah, another really, really good question. And we did do water sampling in the river and of the fresh water in River Darwin. Essentially, there was very little evidence of pharmaceutical pollution in the river, which is partly why we chose that river. We did find very, very low levels of tramadol in the river, in the sort of 1 to sub 1 nanogram per liter range. But really importantly, we didn't find any evidence that this was actually accumulating in the tissues of our control fish, meaning that it was sort of unlikely to be having any sort of actual effect. The only other thing that we found in this river, and to be frank, we find in basically all rivers we sample in, is caffeine in astronomical numbers. And indeed, there's sort of some recent talk, and I think a recent article came out about how it's actually very hard to find a control condition in ecotoxicology these days, because most environments you go to have at least one, if not more, different sort of anthropogenic substances in these systems. So yeah, we did check that.
That's crazy. So then Jean-Noël is asking you, so first of all, you say very interesting talk, thanks.
Thank you.
I heard a lot about endocrine disturber. Do you think these pollutants could have such effect on fishes' behavior as well?
Yeah, definitely. So endocrine disrupting chemicals are really one of the classic cases in ecotoxicology. And I think, there was famous experiments from Canada in the early 2000s showing that components of the contraceptive pill at very, very low concentrations were actually feminizing fish populations and leading to population collapse. And these are sort of concentrations reflective of what you would be expected to see in the environment. But also a bunch of actually collaborators on this paper, including Michael Bertram, have done lots of work on how anthropogenic endocrine disruptors can affect all different types of behaviors and also physiological and sort of growth traits in fish as well.
Okay, so I think I will just read one last question, but there are more in the chat. So to the people that didn't have an answer yet, I encourage you to go to the Discord in the channel of Jack and write your question. So the last question I will read is from Martin. Cool talk. What do you think is happening to these fishes with the clobazam? Is this affecting their perception, their motor locomotion, or they forgot that that their friends are friends?
I really think, and again, this is a little bit of guesswork on my part since we don't have really solid evidence here. I suspect it's having something to do with their physiological stress and how they're perceiving risk. So we haven't found any evidence. These doses aren't high enough to really affect motor coordination or locomotion or anything like that. And we're not seeing sort of differences in swimming speed or anything, or even directionality of locomotion. But we do know from lots of previous studies that it does affect physiological stress and risk perception. And so I think that's having a flow-on effect to social behavior, and that they're perceiving things to be less risky than they are. And often in fish, schooling and social behavior is a common response to predation threat. And that it's these sort of behavioral dynamics that are then influencing dam passage and then subsequent migration outcomes as well. But essentially, we need follow-up experiments on that.
So there's still a lot of work to do.
Yeah, fortunately or fortunately.
Always. Very interesting. Thank you again, Jack, for